So this week, we are going to revisit our numerical examples in matrix decomposition, especially in SVD. And then we also will have introduction for the spatial transcriptome analysis. So let's start with the small example we used in the lecture. So please implement this input matrix, this A. So you have 1, 2, 3, 4, 5, 6 in MATLAB. Oh, by the way, I always keep updating the toolbox. So if you reinstall to overwrite in the old one, that will be a good idea because you have updated the function. Some of this, such as meta visualization, I already updated. So with that, let's make this matrix example 1, 2, 3, 4, 5, 6. Okay. So this is not a squared matrix. Can you do eigen decomposition for this? It's not because it's not a squared matrix. So if you really want to do eigen decomposition, what you can do is to multiplicate by its transposition. Okay. So here you make a squared matrix, but that is a covariance matrix, something like that. Or you can do the transpose and then multiplication. In that case, you get a 3 by 3 of matrix. Otherwise, you have a 2 by 2. You can do this eigen decomposition for those to get eigenvectors, eigenvalues. Okay. But in general, you don't have eigen decomposition on this original matrix. But what you can do is to do SVD. So if you have SV and SVD, this should work. Okay, so this will give you the U matrix. It's a rotation matrix in 2 dimensions, and then another rotation matrix in 3 dimensions. Connect in the middle is this S vector. It's also a matrix, the singular values. Okay. So what you get is should be the same as our example here. Okay. You get the same values of these 3 matrices. Okay, so the product of these 3 times S multiplied with V transpose is your original matrix. Okay, so you have this. Then we can start to see, okay, can we use truncated? We only use this part, okay, the first column and the fourth element in S, and the fourth column in V. So if I do that, it's U, the first column times S, the first one, V, second column. I'm not so sure for the treatment for the last matrix, whether it should use a row matrix, row or column. Let me try which one is better. Looks the same. Yeah, so I think this is the correct way to use. Okay, so this tells us we can use a truncated, use the major components to reconstruct almost the same matrix of original. But you cannot use the second one. So the second, if you do 2, 2, 2, this will not be approximation for the original matrix because you are using the less important components to reconstruct. So to give the better resolution, you can combine these two to get the original. Okay. So this equation is the same as what I'm writing. Use O. So this expression is just separate first component, second component. Well, this one just compute at the same time. Okay. In this small example, there's only 2 major components really play an important role, because if you look at the S, there's only 2 values in this diagonal position. And the relative contribution you can compute, say, okay, in total, and this may be 98% of the variability in your data can be explained. Well, this one only take, okay, a little bit of portion. So you have a relative contribution for major components and minor components. Now it's your job to come back to this small example. Can you do this numeric example to construct this original matrix and to do the SVD to get this? Okay, you can type this in as an input matrix and play with SVD and get see whether you have this. I don't know how many. Yeah, so you should have a much larger matrix. And this is a truncated version displayed on the screen here. You have typed that in because there's no way you can get the data in there. Mm. Can put the 3 dots at the end in order to do this return, allow the code to start from the next line. So this should work. And so I did this construction of my input data. It's very easy. So any type of this matrix, you can do SVD. Even the complex number in there, you can still use it. Let's see. Type that in. Good. Okay, let's see. So if you achieve this, you can take a look what's inside U, what's inside S. I just click those variable name and look at. Or you need a space between. You need a space between those numbers. Yeah. Right. Yeah.
Sir,
yeah.
When you input your matrix, is there a particular reason why we need to set it up with the
3 dots?
Yes, sir.
I just want to keep the flow of this code, but you can click return to keep going.
Okay.
Otherwise, if you don't have 3 dots, if you click return, it start to execute the code.
Okay.
It just help you to start a new line.
Oh,
Yeah. Yeah, continue new line. Yes. Yeah, you can bring down. Continue. Yes. I think the problem is, yeah. Yeah, you can continue type the second row.
Can't cut and paste it. Right?
Cut and paste.
Just have that. I already typed it in once, so I was trying to cut pieces
Oh,
up, but once you just have to input it again.
it's possible. It's possible.
Oh, okay, I see.
It's possible.
I'll just type it.
Yeah. So yeah, you can type the whole row, but yeah.
Yeah, I put it in.
Yeah, I know.
Okay, I see.
Yeah. Now you know
No,
what's
it's
going on.
the dots.
Yeah, put the dots in order to wait. The 3 dots has to be connected with no space. Yeah, no space. Yeah.
Oh, okay.
And return. Yeah. Yeah. Lowercase SVD. Yeah. The function is lowercase. Yeah, good.
Oh,
Yeah, you just need to set A at the point. Yeah. Instead of like this matrix.
Okay, so you have all this. Okay, so if you have this A matrix, you can use this called image SC to look at this. You see this the values. Okay, so when the value is high, you have this yellow, this 2 box here is 5, 5. When value is 0, you have this dark blue 0. So you have this. So this is a way you can visualize the matrix. Use some kind of heat map. And then you can change color map to jet, to different options. You can see, okay, these options you can use. Now, if I want to look at image, I see my U matrix. This looks like this. Color map, default. So if you have trouble to figure out what this shape looks like, you have this. So that's your U matrix. And then you can have image SC for the V matrix. The V matrix is 5 by 5. U matrix is 7 by 7. S matrix is fill up the zeros. It's only first two components the most important. Okay. So what we can do, we just keep the top 2 components. The rest of them are not important at all. So what we can do is to reconstruct the data, use V top 2 components as 1 to 2, 1 to 2 times V, same 1 to 2, but remember to transpose it. So we can just keep top two components to reconstruct the whole original matrix. Okay, so that's what they, so this is the expression, keep top two components. So those values are the ones they displayed on the slides. Although this U matrix is 7 by 7, so the rest of them are not very informative because depends on this strength matrix, there are only top two are useful. The rest of them, because the zeros in there have been, when you do the multiplication, they're all gone. So that's the way you can compress information. So the whole matrix can be huge, but you only need this very skinny two matrices, only two columns to represent the whole structure of your original data. How I show the color map, so I use a function like this called image SC, then put your matrix in there. Oh, you mean the color bar here? This one here? Which one? Oh, color map. Okay, how I see the options. The options are like by the tab key. So color map, then you have a space, and then you press tab. The tab key give you the options of what you can have. Okay. Yeah, so how do you select this is important. The sum of this color map support this continuous variable. For example, those ones are good for the continuous variable change. So you have a continuous variable change from a negative 0.8 to positive 0.6. So you should use this kind of a color map. You don't want to use those like flag. This is only for the categorical. So you see, this will be not good for you to do those, to show the continuous change of the values. So just remember, some of those are for the continuous variable, some are not. So with lines, that's not good for continuous variable. It's good for color different categories of the discrete numbers. Okay, are we all right with this? Okay. Now we are going to do an image decomposition. So now we played with the small examples like this. Now we are going to use slightly larger matrix, close to what you have for the whole gene expression matrix from single cell data. The one I'm going to use is from this example. I think it's a good idea if you type this. No. So this will read image file and then convert the color picture to gray, black and white with grayscale, and then show the image. So let's do this. So I'm going to clean my space. Close all. let's just read that information. So this is a matrix. If I use image show, and this will give me this picture. This picture is, let's see, what's the A's dimension? So this is 3D because the color is in there. So this matrix, it's not a matrix, it's 480 by 640 by 3. 3 the indicator of the different color combinations to give you red, blue, yellow, something like that. So we need to convert this. This is a tensor, so A is tensor. So that's why you have a color here. So we need a function to convert this A into 2D. Okay, so that's a B. What's the function of the B? RBG to gray. Okay, to gray. RGB to gray, and A put in there. So this function to get rid of all the colors and to give you a matrix. Now you can view this matrix here. Now, if you do this show again, okay. So in this matrix, each position is give you each pixel's grayscale color, not color, but the strength. Okay. For instance, if I replace all this to some values, I will show the different color, different grayscales in the picture. For instance, I replace 1 to 100, 200, and column 1 to 200 into 256. Okay, I replace all the values because this integer with 8 bits, so it has a ceiling, it's 250. Now, if you plot this, you will see this space has been replaced as a white color. Okay, so basically 250 is. So you can manipulate this matrix. I can put 100 to 0. So this, it will show a black box, right? You get the idea? Okay, the values you can change in their coordinates. Now we are going to do the SVD to this A. So we need to convert this to double. Okay. Because A was stored as an unsigned integer with 8 bits, so it saved the memory because you don't need a double position because there's just integer to save space in the memory. But this function only take double, so we need to do a conversion in order to get those values. Okay, so this is first step. We already show the original then we are going to do this. So they use a different version of this SVD, but the same thing, okay? Let's call it the same variables. So let's call them. Let's call it S1 even. Okay. Now you just multiplicate them and then convert the back to the integer. Okay, so this is a use decomposed components, put them back, and then convert the data type to integer. Then you can view that again. This is constructed. This is reconstructed. You don't feel anything because the whole information of the original matrix has completely reconstructed. Use all the major components. So if you compare this picture, it will almost the same as original. Now I'm going to look at this S matrix to see its diagonal position, what's the value. So the component 1 have a contribution at this level, and then the second component this level, so gradually change. Looks like I can use, still very informative until that's a lot. So let's do this figure and the image. Let's see to look at this S1 matrix. I want to find where, well, it's huge. No, nothing. Let's do this figure. Extract the diagonal position of S1 and plot its value. Can anyone tell me what I'm doing here? Anyone knows what trying to do? I try to. Yeah, so highest value will be the first one. So highest value is this one here. Basically, I want to look at this curve because the first value indicate, okay, what's the contribution for the first component and what's the contribution for the second. So I just follow these values, and these values are in this blue curve here. And I try to find a position where maybe, for example, in 24, 24 position, That means the top 24 components. After that, the contribution for the rest of the components will be looks very small. Okay, so I want to find the cutting position and where I can keep how many components in there. For example, I will choose 12 first. Okay, I believe 12 and 24, no much difference, but it make this matrix much small to rather than 24 or even to 500, not very useful. The rest of the components are not so important. So I will keep top 12. So to reconstruct, use the top 12, I will do a new one. Okay, only use 1 to 12. This will be difficult to write. S, I need 1 to 12, 1 to 12. And the V, I need column 1 to 12. So in this way, I reconstruct my original picture matrix. I only use top 12 components. Let me check. So what's going on here? I think that's a problem. Okay, so this should work. Let's give you some time to type this in. So remember, and then I can look at this reconstructed the figure to bring up a new window. So that's my reconstruction. Seems not enough. Okay, 12 also gave you some major components, but not enough. So maybe we should try replace this to 24 or even more. But you get the idea, okay? So I will go around and check whether you can achieve some of this. Not yet. Not yet. Okay. Yeah, so let's do this. I had a typo, so get rid of it. It should work. Yeah. No, not working. Yeah, I had a typo which you didn't fix. The typo is here. Delete this. This should work. Yeah. Yeah, you can replace because you use the U instead of U1, so you can use your own variable names as the same. Yeah, this should work. Yeah, you should do written. It should work. Yeah, good. Yeah. Yeah, I had a typo in my original code, so. Oh, you use yes.
No, I was just checking different numbers. But
Yeah.
After the call,
Yeah.
it looked really blurry.
Yeah. So maybe try 24 or something else. Yeah. But the major components already reconstructed. The building.
When you change the numbers, does that have to be consistent throughout the each?
I think so, in order to make it possible to do the multiplication. Yeah. So now you can imagine if you replace this straight picture to gene expression analysis from a matrix, like you get it from single cell, it will be the same idea, okay? You can learn these major components in your whole matrix.
That means we learn the major genes.
Yeah, it could be matter genes, could be major clusters of cell types. So you have to look at the values in U and V matrix.
U matrix contains the information.
U matrix contain information about the matter information for the rows, and the V contain information for the columns.
That means that those should be the cells.
Could be the cells, could be the genes, depends on your input.
Because I think when we do that, we use the transpose.
Yeah, transpose.
The gene should be the column.
Yeah.
Column.
Yeah.
That means the U should be the cell cluster.
Yeah.
And the V should be the gene.
That's right. Yes. Yeah. SVD, you don't worry about the transpose or not. So any shape you can put in SVD. Okay. Okay, good. So this SVD sketch function allow you to select, I only use top 15, or you can put different options. Right now we are doing this full SVD, so we have to manually truncate. But this function just some built-in features to allow you to say, I only use top 15, and then to construct, and you get a picture like this, okay? Are we okay with the SVD? Done? Okay. Now let's clean the space. Then we play with the ICA example, then we have a break. And to clear all, close all. Now, if you have access to this Google Drive, and you will find a zip file called PCA ICA. So you will have to download this. And then find this file in your download folder. Downloads. You need those. So this is a zipped file, so you need to do extraction. After extraction, you go into the folder and find demo ICA #1. So that's what we are going to use. So if you open this, you can click this button to run. It will allow you to change the folder. Once you have this window, you just follow the instruction to change the folder to allow this ICA to run. You will get this, okay? Change the folder if you're in this, yeah, you just click run. Yeah, it will change the folder to help you to. Okay, yeah. Yeah, you can go ahead and click play, but maybe it's very noisy, and I hope, yeah, lower your volume before, otherwise the whole room will have this noise. Oh, there's no.
I think there was problem with Mac.
Oh, with Mac. Okay, that's fine. Oh, that's unable to play video because. Oh, I will try. Okay, that will be a problem. So the reason is those signals are from audio, and so let's see if I can play here. Otherwise, I don't know. Oh, now no speaker. Yeah, so I have some problem to hear any of this.
The Guardians of the
Who got the sound? Okay, can you play? So you play #1. So #1.
The guardians of the electronic stock market NASDAQ who've been burned by
So #1 is a news, and the #2, the true. What's this? Okay. #3. So this 3 will be mixed in different portion and then become a mixed #1. You change the parameter and then you have another mixture information #2. So relative portion of the 3 different tracks give you different contribution, and then mix 1, 2, 3, and you give this information to the fast ICA, and the fast ICA use particular algorithm can reconstruct #1 sound. It's reconstructed from a mixture, okay? And #2 is also constructed. And the number 3, so 3 different channels, and the information has been recovered completely.
The guardians of the electronic stock market NASDAQ who've been burned by past questions are moving.
So these 3 are using one of the algorithm called the negative entropy. And another algorithm implemented in fast ICA is called kurtosis, and you can see its results to separate 1, 2, 3 source.
The guardians of the electronic stock market NASDAQ who've been
Yep,
burned by
it's
past
good.
questions are
Number 2. Yeah. And then the last algorithm didn't do a very good job. If you play this KICA algorithm, results to, yeah, it's not completely separate the background music from this. Number 2, it's supposed to be pure music, but there's some noise still there. Number 3 should be that news report.
the guardians of the electronic stock market NASDAQ who've been burned by past ethics questions are moving.
Yeah. Okay, so this is just a demonstration, and their source code is inside this demonstration, but the different algorithm play for this process behave a little bit differently. Okay. Okay, so it's good or bad that you cannot play with this sound, otherwise the whole music play around. Anyway, so let's take 5 minutes break. We will come back to work on the spatial transcriptome. Okay. So this is spatial transcriptome is a suite of technology, but we are using 10x genomics platform as an example to show you what you can achieve. So you will have a tissue, the slide section, and then take a picture, and then you have this probe to sample at a different position on this slide to get barcode information for gene expression for all the gene expression in this particular spots. So you have the physical location of each spot, their relationships, then you have this profile. Let's see how this works. Noise. You see, once you have this profile, you can look at the different genes expression and their physical location on these different spots. Okay, each spot has thousands of genes you can work out, and you can look at the set of genes, see whether they always highly expressed in this corner, and what this structure is, what are the feature genes which highly expressed in this part. So a lot of questions you can ask. Okay, I have on Twitter, and recently this particular group, they use spatial transcriptome to do this, use mouse model, ischemic brain injury, so they can reconstruct some picture like this. Okay, they have an animal model after this ischemic ischemia, and then you have at a different time after the injury and what's the gene expression profile in the local brain area. This is one of the applications. Then you can have dynamic picture of what kind of gene expression program across the time on this particular position of the injury. So this is one of the examples. So let's see what our solutions for this kind of analysis. So you have to download another package. Let's put my name here, GitHub. In order to find the repository I have made. GitHub is where people put the source code to share with other people. And that's my username. So if you're on this page, it's correct, then you click repositories. And the second on this. I only made this project public because of the teaching, okay? I'm not ready to share because this is an ongoing project, but then let's click into this repository and you browse this down and you can copy this quick installation. So once you have this, you can come back to your MATLAB and close all, clear all, and find the place where you want to install this package. I will go back to this folder and to paste the code, okay? So let's see whether you are in the, so I want to install this side by side with the single cell toolbox. Of course, you can install in the download folder, it's fine. Get it? Okay. Not yet. The next repository is spatial transcriptomics. Then you browse down and you have this. Okay, good. You can just click this to go back to the original. Yes, that's place. That's good. Working, okay, good. So remember, you installed everything under this folder, but it's fine. Yeah, yeah. Okay, it's good. So you're done. Yes. Yeah, yeah. So if you don't switch to the right folder, it's fine. But you remember you installed under this folder.
Okay.
Yeah, but yeah, that's fine. Yeah, no problem. Yeah. So what you can see, the folder and the current folder is here in this region. But we just do this one-time use, so it's fine if you install in under the ICA examples. Any folder should work. But if for later use, you probably want to install this side by side with another toolbox so you can keep using. Okay. Everybody is done. So we're going to do next. Now, if you have this, then you can type STGEA2. Okay. It was SC, but now it's ST. ST stands for spatial transcriptome. Same interface, you can load a processed example and see what you get. I hope everything is fine. Take some time. Getting anything? Okay, good. Yeah. Not yet. Yeah, you can go ahead and load a process example. Process the example. Yeah, good. Yeah, feel free to go ahead to load the example, process the example, then you can have some quick idea of what's in there. So I just click this and to get processed. Okay, if you click this button, you can switch between 2D, 3D. And those buttons you can click and change color, something like that. Okay. So on this picture, you have this tissue slide image, and that's your profile for the probes you sampled, all the spots in there. There are 1,175 spots, but we can trade those spots as individual cells. Okay, although they are not, they are mixture of maybe 5 to 10 cells under each spots. So if you treat them as single cell, you can do all the SC gene expression analysis, just clustering, cell type identification, just as single cell. But now they have physical location, so you know where they come from, these spots. So that's a difference. Okay, yeah, it's good. So now the color by default is a library size. If you sequence more UMI, so you have higher numbers in this spot. So some location you sequence more by chance or by the nature of this particular tissue under this spot. You can click this button to hide either the image or so you can switch them on and off with this button here. So if you zoom in, sometimes this spots looks too small, you can change the size. Okay, make it larger or small, you know, for your visualization purpose. It's too small, and you can double click and to increase size. So anytime you can export this to PowerPoint. To get the presentation view to share. Okay. This is to change the level of transparency And then you change different color map. Okay. This is not good for continuous variable. Again, so some is better than others. Okay, so then you know what those buttons are. These are the to show gene expression and on each spot. So you can select this and to get the sum of the genes, for example, those example genes. Then click OK. I should have their expression profile on their distribution on this slide. Okay, those genes are just randomly selected. Some of those are informative compared to others, some are not. So for instance, this gene tend to express at this boundary here, okay? So you have a chance to check all the genes expression profile. This gene highly expressed in this particular corner. Okay, so close all other figures. You can do the same thing as for single cell data to do some kind of filter and see you want to get rid of gene lowly expressed. It has to be 7 or 8% of the spots have to express that gene. Then this will remove some of the lowly expressed genes. Okay. And then you can trade all these spots as a single cell. So you can do clustering. And for example, this already been done. And you can use t-SNE to put the cells in a 3D space, and then to do k-means and use existing. So this spots has been classified or clustered into different categories based on their 3D t-SNE embedding. And then you colored different clusters. And we found, for example, this purple cluster match the structure of this particular, I don't know what it is, but the cells look at spots within this structure have the same expression profile. And the boundary, this orange one, the cluster number 2 is in this boundary here. And there are yellow ones in the top tips here, and the green one, some in this region, some in this region. Okay. And there are other ways to do the clustering. Instead of using embedding, remember, we can use SC3 from the expression matrix to do the clustering, the same idea, and it's already been done. So you see different ways of do the clustering, slightly different from the embedding. But the major structure, and you can refer to this slide image to see, okay, it makes sense to me. And then you keep that clustering. So once you have clusters, you can see, I want to select one of the clusters, for example, the yellow ones, and highlight those spots. And then see, I want to look at the marker genes in the brushed spots. So you can see, I want the top 15 highly expressed gene in that particular spots. I selected. Then you should identify those genes highly expressed in the region you selected out of this 10,000 genes. Same function I use in the single cell data. Okay. So once you know, okay, those genes, collagen, and you get some idea about this collagen tissue part of this. There's at least 2 collagen in there. So oh, it's using regression. Yeah, regression.
Compared to all the other areas?
Yeah. So if you set the select spots have a 1 and all the other areas has 0, so you want to find the gene to explain this 1-0 difference. Okay, any questions so far? Fine, okay. Some buttons should work, some should not. I haven't finished this, okay? Because and you can do the same thing for different clusters. To find the marker genes for all the different clusters by cluster ID. Using different methods. Maybe I try this. So those functions are the same, borrowed from single cell data analysis, but apply to here in the context with the spatial information of all the spots. You can also assign cell types for each spot. Okay, in this case, we have a list of suggestions. What's the top possible cell type? What's the second? What's the third? So that information will be useful. You can see, okay, this part maybe is a mixture of cell type 1 and 2, the 2 different cell type. Okay, so this is a marker genes for cluster 1, 2, 3 to 6. Okay. It's a busy picture. But you know it's the same as a single cell data, okay? So for cluster number 6, which is a light blue, and we have marker genes RRBP1, KNX3-1, so those are marker genes for this And for number five, we have a lot of marker genes. Number 5. Okay, so you can do some very basic. There are more complicated analysis. I will talk about them later, and we don't have enough time today. So. Well, I don't know. I don't know. We have to read the original design of the experiment where this data set is obtained. I didn't pay attention to this. Yeah. Yeah, you can use that. You can also use a frozen tissue slice without the stain. That's from the. You can use either fresh frozen or use a. I think you are referred to FFPE. Yeah, you can use both. There are a lot of discussions, tutorials online, and in the field, there's hype. People want to do this to understand the different cell type interaction with the spatial information you will get. For example, solid tumor and their periphery tissue and the cell type microenvironment to be described more accurate than dissociated cell type single cell analysis. Now I'm going to show you how can you download the data and process it, because this one is already aligned. So the distribution of your spots profile aligned very well manually. I actually aligned them. So the raw data you have probably not aligned. So I will show you an example and how can you do the manual alignment. Okay. So let's start from this two. Okay, not SC, ST instead. ST, then we will download from geodatabase a data, for example, this one. If you want to know more information about this sample, you can Google and find the geodatabase record and see, okay, which experiment, what sample, what animal, get what purpose of the experiment. So this is one. You see it's not aligned yet. Okay, in this case, I have to switch this on to have this manual alignment. If you click this M, it's allowed you to manually move your spots. And you have to maybe have to do the flip, I don't know, rotation. So I did a flip and a rotation, and to match. And then probably I have to zoom a little bit. Yeah, it's very difficult sometimes. And once you finish, you can save the file. Next time you don't have to do that again. Okay. Yeah, so this is easy one. I have more complicated. Sometimes you never achieve 100% accurate, but you just give some reference about which part of this tissue contribute to these spots. So it doesn't matter actually. Yeah, they do have special barcode. I don't know how to use that information, so I just manually align them. Yeah. Yeah, somehow something like this. Okay. Now I'm done alignment. I have more complicated examples. I will show you the one I'm going to use again. So this is from a database. So this is a spatial omics database. The one I'm going to use is one from this study. All I need is a sample code. So I want to use GSM. Let me type that in. So I want to use this GSM 5726156. So we're going to try this example. This from a mouse in testing, so the structure is more complicated. Okay, it's a challenge to align this. So I'm going to switch off this and do a session number here. I use this one. Okay. Let me clean the window so you can see which sample I use. This ID is you will see the ID is this one here. If you want to follow it, follow it. If you don't have time, just watch the structure, the process. So you can get those numbers from other people's publication, because once they upload the matrix and also the image of the slides, it should be in the geodatabase, and you should be able to download and automatically process. Okay, look, this is more complicated. Okay. You have to be very skillful to align this. If you have this, give it a try. Again, the ID is, this is ID. This is ID here, okay? ID is here. See whether you can find a way to align this perfectly with this picture. This is just one time. Once you finish alignment, you can save the file. Next time you don't have to do it. Okay.
We cannot.
Oh,
Because you just mentioned each time I will install, I use a very long name.
long name, yeah.
You said you just still the MATLAB. I think maybe that's
Okay. Yeah. So I make those spots smaller so I can see through, and then we'll do a rotation, see whether I need of this. Well, I think this spot here should match to here. So I'm going to rotate that again to allow, no, this one, maybe I should flip it. Okay, so this looks like this. So I did some rotation and then flip image, mirror this flip. And then move to here. It should also look at those space. That's the one. Yeah, I believe this is a correct alignment. Not perfect, but the ballpark should be something like that. So this is raw data. I haven't processed it. What I'm going to do is just filter out some genes. This 31,000 genes, that's just too many genes. Let's put 8%. Okay, now I have 8,000 genes. I can do a quick clustering algorithm. This will be T-SNE based. Let's have 5 different clusters. Let's see what's the cluster we can have. This structure has a circle in there, so I hope some of these clusters will be around the circle region rather than just by distance. So we are not using any of this distance in our clustering algorithm. So the algorithm will treat every spot as an individual cell. So that will be a T-SNE, and you can imagine. And then to find those clusters, 5 different clusters that map back the cluster's ID with different colors in there. So you see some of this this orange clusters on the circle, this structure. Okay. Yeah, as I said, there are a lot of functions which are more complicated ones under development. Some function like we will detect the boundary spots and look at their interaction. And their interaction, if they are based on the physical contact, will be, suppose those kind of interaction will be stronger compared to the spots which this one against that one which not physically contact. We try to detect that information. So this is just one way we utilize those spatial transcriptome information that you can imagine if you have a real project, what you can do with this type of data. Okay. Any quick questions? Are we okay with this part? Okay. We can compute some of these feature scores for each spot. So these scores will be, each spot will compute a number. But this number is come from the synthesized information from multiple genes. So if you collect a set of genes from the same pathway, so that information will be integrated and compute for every spot. Some spots maybe have a high values indicate the activity about this specific cell or spots have a high activity regarding to that particular pathway. Okay, so this is like a pathway based. So you have a signature of the genes. You can select. I don't think this human. So you select all this. Let me just randomly select one from a curated gene set. There are a lot. I think I should find one which Which one? Some epithelium cell markers, maybe. Okay, let's try this fiber formation, and then we'll see which spot on this structure have more involved in this pathway. Okay. Same here. Okay. So the higher value means reaction of the activity is higher for this region. Where is the menu of this database? Specific gene expression. I just did this score. Yeah, this one here. Yeah. Let's see. Maybe ontology of gene set. I want to find. Okay, I want to find some epithelium cell development, maybe. Okay, let's see this. Which one? E? I don't know which data this database. Yeah, you can certainly have. Let's try marker genes. Mouse. There are many possibilities. Yeah, so that's my score. Yeah, I try to think this makes sense because if you look at where this particular score is higher, it's in this region. It's supposed to be epithelium cell rich region. Yeah, it's hard to see. Okay, so that's all for today. All right, I'll see you next Tuesday. Maybe it should be endothelium.